Who were the Incas? Do elephants ever forget? Are elephants afraid of mice? What is this book?
This book is called Still More Tell Me Why. It was one of a series of books, Tell Me Why, More Tell Me Why, and Still More Tell Me Why. And it was really a science book that I would have spent a lot of time reading as a kid growing up, somewhere around between 10 and 12. I'd find a quiet corner. I can still see where I was in the house and hope nobody would find me. There was jobs that probably needed to be done that I was hiding from. But it was, I would literally, find time, love to kind of get stuck into it and just read. And it was just one of those books that really fascinated me. And in particular, there were parts of it, the ones I always tended to veer towards were descriptions on disease and how disease is caused. And so there were some I would have been always up at this end of the book. I was just fascinated by it, fascinated by what caused disease. I would have had there would have been nothing that would have triggered it from that point of view, but just had a real interest in not just disease, but the other simple things like why do humans have hair, when do you stop growing, things, one of the things that's in it, I won't be able to find the pages, what is ESP?
And what is ESP?
Well, I think it doesn't exist. But would have been a thing, I think, around the 70s in terms of, extrasensory perception. So my name is Siobhan McClean. I'm a biochemist, and I'm in the School of Biomolecular Biomedical Science in UCD. And I work on cystic fibrosis infections and also vaccines, or trying to develop vaccines for bacterial infections.
Well, that's kind of topical, isn't it? Or it certainly has been, this word vaccine. We've all heard the word vaccine, I'd say, in the last 2 years more than we've ever heard it before.
Yeah, there's never been a better time to work on vaccines. It's been very interesting. But so we don't work on viral vaccines, but we do work on bacterial infections and trying to get vaccines to protect against those. But there's never been a better time to work on vaccines. It's something that everybody kind of has some idea about.
What is a vaccine?
A vaccine, to put kind of simply, it's kind of training the immune system or telling the immune system this is something that you need to protect the body against. So it's like exposing the immune system to a portion of this thing that's going to cause the infection, and so that when the immune system sees it again, it goes, aha, I've seen this before, and then is able to has the tools to mount a response to protect the body from that. Before the infection actually takes hold. So it's usually you're trying to develop a vaccine to prevent the infection, but often actually we'll try and get a vaccine which the person might still get the infection, but it won't be as bad. So it's kind of the immune system kind of kicks in and
modulates it.
So it doesn't do its worst.
It's not naive. It's not seen it for the first time.
Yeah.
And we have a couple of sort of very sciencey looking things on the table, but these are not particularly sciencey. Are these actually things that you would use in the lab?
Yeah, so this is what we call a gel in the lab. It's a little bit old, so which is why it's got this beautiful curly edges. But this is how we look at proteins in the lab. So when we're trying to develop a vaccine, the vaccines that we develop are proteins. So they're molecules in the body that do work, that actually have effect in the body or in the cell. And so what we do is we look at it. We'll pick. See if you can see this this big band here. I don't know if that's visible on the video.
Maybe it'll be easier on this one.
So one of those, what we call a band, will represent a vaccine antigen. So in other words, it's the molecule that we're going to want to use as the vaccine, which will again stimulate this immune response and protect people. So what we do is we look for particular molecules in bacteria, these antigens or vaccine antigens in bacteria, and we use those we try and develop those as vaccines.
And you make that sound so easy. So once you found the molecule that you want to use, how do you actually then turn that into a vaccine?
So firstly, I suppose that we figure out what these molecules are, because they're the molecules that the bacteria use to actually stick to human cells. So that's our first step in the process. We try and find the molecules that bacteria use to attach to human cells. And then once we have those, we have to go back to the genome of the bacteria, take the gene for that protein, put that gene in another cell so that other cell makes lots and lots of the protein. Then we purify the protein, and we then, when we have the protein in a pure form, we test it, and we test it in mice. So we'll take groups of mice and we'll give the mice 3 of these immunizations, and in the same way as people got three COVID-19 vaccines. And then we'll come along, we'll give the mice the infection, and we work out if the vaccine works, the mice won't get the infection, and if the vaccine doesn't work, the mice will get the infection, and so that project will stop, and we won't go any further with it.
It's actually it's reasonably straightforward.
It's a reasonably straightforward process, from start to finish. It's once we know what the protein is, we have to figure out we have to get the gene and then transfer the gene and purify. Now it takes a long time.
We've described it there in the space of 3 minutes.
Yeah,
obviously,
it takes a long time.
more complicated than that, and you've made it sound very simple, which I know it isn't. And the reason that, so that's probably why the syringe is here. The reason that you test it on mice, I can imagine people will say that's not right and we can't be doing that and all the rest of it. But the reason for that is...
Well, we can't test on humans, so you have to test it. So the mice is actually a very well understood immune system, and so we can test them on mice, and we know if it works in mice, it has a chance that it'll work in humans. Often things work better in mice than they do in humans, so even if it works in mice, doesn't necessarily mean it'll work in humans, but it's the best chance we have of knowing before we test in humans. So we have actually one vaccine that we're hoping to actually test in humans in the next year. I'm hoping. Cross my fingers, and it'll be a very small trial that we'll do in maybe 10 to 12 people, and it'll be very much looking at the safety of it, because it has to be safe in people. So we start very, very small with only about 10 people. So I know a lot of people wouldn't be uncomfortable with the fact that we have to test things in mice, but there's no other way of knowing if it works without actually testing it in an animal, because you have to actually have the whole living mammal with a liver, with lungs, with the various organs.
Exactly.
Yeah, so that's the only way we know it works.